Good morning, everybody. Just going to pick up where we left off on Monday. We were working our way through the various lines of defense, and we first got to learn about the first line of defense, and that included your skin, some of the mucous membrane, chemicals, and other stuff. Now we're going to move on to the second line of defense, which, based on its name, tells you right away that it is number 2 in order once the penetration of the physical and chemical barriers occurs. Okay. The first set of structures that we're going to talk about are these antimicrobial substances. Okay. We have 4 different antimicrobial substances that are part of this second line of defense. First, they're called interferons, which the beginning of that word kind of tells you what it does. It interferes. Okay. How it interferes, we'll talk about here in just a second. Okay. But these are proteins produced by lymphocytes. We know lymphocytes are those major soldiers of the immune system. They're also produced by macrophages, which, remember, macrophages I always think of like Pac-Man running around and chopping stuff up. And then fibroblasts, which are connective tissue cells. But they are proteins produced by those different types of cells that have become infected by a virus. Okay. Once released by the virus-infected cells, these IFNs diffuse into the uninfected neighboring cells, or diffuse to the uninfected neighboring cells, where they reduce the synthesis of antiviral proteins that interfere with viral replication, which is what we're showing off right here. So here's our virus infecting a cell. Doesn't matter what the host cell is, if it's a lymphocyte or macrophage or a fibroblast. Okay. The interferons are being produced by that host cell that then travel to a neighboring cell, and you can see what happens. Okay. The interferon binds this receptor on the neighboring cell. Signal is sent to the nucleus, activating genes for antiviral proteins. Cell produces those antiviral proteins, blocking viral replication if cell becomes infected with a virus. Basically, it prevents this from happening, which is what most viruses are going to aim to do. Basically, take over gene expression inside the cell, make more viruses. This prevents that from happening. Okay? So they interfere with viral replication. If you want to think of it that way, the very last 4 words there kind of tells you what interferons do. The complement system is a series of proteins that are normally inactive. Okay, there's generally somewhere in the neighborhood of 30 or so, found in our blood plasma and plasma membranes, produced by our liver, and when they are activated, think of them like dominoes, where one knocks down the other. And that's why we call it the complement system, because they complement one another, not in like, hey, good job, but they help activate the next one. Okay. So they work in kind of a cascade of events, and they destroy microbes by causing phagocytosis. They perform cytolysis, which the word cytolysis, cyto means cell, lysis means to break down, so it causes the cell to break down, and they're involved with inflammation. Okay. Antimicrobial proteins, or AMPs, are short peptides that have a broad spectrum of antimicrobial activity. They can attract dendritic cells and mast cells that have, or that participate in immune cells. And then iron-binding proteins do exactly what it sounds like. They bind to, or they reduce available iron by binding to it, which iron is essential for bacterial growth. So that's how they can prevent bacteria spreading, basically. Okay. Questions on any of those 4? All right. The other 2 components of our innate immune system include these natural killer cells, which we learned the other day are lymphocytes, and then phagocytes, which there are 2 major types of phagocytes, neutrophils and macrophages. Macrophages, which we know develop from monocytes. Like I said the other day, I wish they call them natural killer lymphocytes. That would make it easier to remember that they are a lymphocyte. These are not the T's or B's. We'll talk about the T's and B's here in just a minute. Those are part of the adaptive immune system. These are part of the innate or non-specific immune system. Totally, they're only about 5 to 10% of the lymphocytes circulating in our blood, but they do, and you can tell based on their name, natural killer, they do kill a lot of things. They have a wide variety of things that they can kill: infected body cells, certain tumor cells, will attack any body cell displaying abnormal and unusual plasma membrane proteins. When they bind to target cells, they can cause the release of granules containing toxic substances from natural killer cells. These granules include perforin. Perforin, here you can see down at the bottom, is this little red stuff. What it does is makes perforations. Anyone know what a perforation is? It's a fancy word for a hole. Okay, like I talked about the other day, if you poke a hole in a balloon, the balloon dies, right? Well, if a hole gets poked into a cell, the same thing eventually happens, the cell bursts. Okay. Granzymes induce apoptosis. Apoptosis is a fancy word to describe cell destruction. Okay. And those are also released by those natural killer cells. So here we can see a natural killer cell taking care of this. I know it's kind of cut off because it looks like the screen got moved a little bit. But it's attacking an infected body cell. Phagocytes, like we see here, once again perform phagocytosis, which is a fancy word to describe cellular eating, so the ingestion of microbes or other particles like cell debris. We know, based on what we've learned previously, that the two major types are macrophages and neutrophils. Okay, they migrate to infected areas in response to infection and then perform the 5 steps in phagocytosis, and those 5 steps are all listed right here. The first thing, and we've talked about this before, I compared it to when my wife makes brownies and I get attracted to the scent of brownies in the kitchen. This is a chemical attraction to a site of infection due to chemicals that are released by invading microbes, damaged cells, the complement proteins that we just talked about, or even other white blood cells. Okay, so that's what chemotaxis is, the ability of these phagocytes to know where to go. Then they adhere. Adherence is where the phagocyte plasma membrane attaches to the surface of the pathogen or foreign material. We can see that here. Basically, it develops these little pseudopods, which are like little mouths that are developing, a lower mouth and an upper mouth, if you will, kind of surrounding the microbe. And then it pinches it inward and pulls it in, and that's what the ingestion is. Just like when we swallow something, we're ingesting it. Here our cell is swallowing in that microbe, and it becomes engulfed in a phagosome. And this phagosome is a microbe inside of a sac, basically. This then allows for lysosomes, which we remember are the organelles that do the digesting inside of our cells, to fuse with it. And that's going to allow for digestion to occur. So inside the cell, phagosome fuses with the lysosome, forming a phagolysosome. Lysosomes contribute their lysozyme, that enzyme that we learned about the other day, kills bacteria, as well as other digestive enzymes that destroy cell walls or degrade carbs, proteins, lipids, and nucleic acids. Phagocyte also forms lethal oxidants, causing a process called oxidative burst. These guys and their enzymes very, very quick in action, killing most bacteria within 30 minutes. Once the killing has occurred, the residual body can then be discharged by producing or performing exocytosis, so that can spit out what's left over, and that's what the killing aspect of the step is, shown there, number 5. All right, there is another form of non-specific immunity that we utilize, a couple of them. And we've already mentioned this inflammation word before. But this is a non-specific defense response of the body to tissue damage. This is non-specific, so once again, we learned the other day, you're born with it. That's what innate meant. And it's non-specific, which meant that it didn't have a memory. We respond the same way with inflammation to a cut as we do to a burn, as we do to a broken bone. Okay. to a pathogen invasion. It's very, very different what's hurting us in those cases, but our body with inflammation responds the same way. Four major symptoms of inflammation include redness, pain, heat, swelling, and then a fifth, which is a possible loss of function. I like to think of one of the easiest examples of this, which is a pimple, which is inflammation in a oil gland in your skin. We've all had pimples before. Most of the time they show up red. And to touch them or you can feel them a lot of times on your face, they feel painful. They're going to be a little bit swollen, sometimes really swollen. What we generally don't feel unless you can experience a huge pimple is the heat that they produce. It's very, very minimal because pimples are very small. But if you have like a broken leg and you have inflammation occurring at a very high level, you're going to feel some heat radiating out from it. And possible loss of function occurs. Well, that would be like if you broke your leg. That's obvious. But like swelling can become so bad in cases like a sprained ankle, your foot might not work the way it's supposed to. So very, very extreme cases where you see loss of function. The function of inflammation is an attempt to dispose of microbes, toxins, or foreign material at the site of injury, prevent their spread to other tissues, and to prepare the site for tissue repair and attempt to restore tissue homeostasis. So if you ever wondered why when you hurt yourself things balloon up, it's because you're sending a bunch of blood flow to that area to try to fix things. Fix the damaged cells that have become obviously damaged. Here's how inflammation works. And because it's inflammation, or because it's a non-specific defense mechanism, and I just stated this, response of a tissue to a cut is similar to that of a burn, radiation, bacterial, or viral invasion. So the 3 steps, regardless of what is causing the damage, involve, first of all, vasodilation, triggered by chemicals released by damaged cells, histamine released by mast cells in connective tissue, and basophils and platelets in response to injury, as well as neutrophils and macrophages that are attracted to the site of injury. And so we want to try to get the white blood cells to the area so that they can fix what's going on. Kinins are also chemicals released that act as chemotactic agents for phagocytes, attracting those white blood cells and macrophages, or the eosinophils, or neutrophils, and macrophages. Prostaglandins are also released by damaged cells and intensify the effects of histamine and kinins. May also stimulate immigration of phagocytes, which immigration is step 2, where the white blood cells leave the blood vessel by poking through the wall. Leukotrienes produced by basophils and mast cells increase permeability and aid in the adherence of phagocytes to pathogens. We talked about leukotrienes just a little bit. Those were local hormones back when we talked about the endocrine system. Different components of the complement system will stimulate the release of histamine, attract neutrophils by chemotaxis, and promote phagocytosis. Other components can destroy bacteria. Once the white blood cells get there, they then need to go to the area where there's actual trauma, like in the case here where we've cut through the skin and microbes have entered. So the white blood cells poke through the blood vessel wall. That's what emigration is. It involves margination, which is where they first stick to the wall, and then emigration or diapedesis, where they squeeze between the endothelial cells that form the wall. Once again, we know this is an attraction to site of injury. We call that chemotaxis, and we've talked about that before. Once there, they can then destroy the microbes as well as dead and damaged host cells, which then allows for the final step to occur, where they can be replaced, which is where the tissue is repaired. We said one of the signs and symptoms of inflammation was heat. Well, heat can be measured with a temperature, and sometimes we describe an elevated temperature as a fever, which is defined as abnormally high body temperature occurring because the hypothalamic thermostat is reset. So it's basically just like me walking over to this thermostat in this room and cranking up the temperature of the room. And then the temperature of the room is going to be maintained at that higher level. Our body does the same thing. Okay? This occurs commonly during infection and inflammation due to pyrogens. Okay? Pyrogens act on the hypothalamus and are inflammatory mediators that cause our body temperature to be reset. The set point goes up instead of being at that 37C, 98.6. We crank it up a little bit. These pyrogens can be exogenous, which means they come from outside the body, like in the case of bacteria, or endogenous, which means they come from within the body itself. So those will be our own body cells. Many bacterial toxins elevate body temperature, sometimes by triggering the release of fever-causing cytokines like interleukin-1 from macrophages, prostaglandins, and histamine from mast cells. Histamine causes vasodilation of blood vessels to the area, causing it to become red in color because of the flow of blood. Histamine also increases capillary permeability to proteins, which allows proteins to leak in the interstitial fluid, causing a local edema. Edema is a fancy word for swelling. And these cytokines also increase sleepiness and cause decrease in plasma iron concentration. So this is why you feel heated, you feel tired when you have a fever going on. An elevated body temperature intensifies the effects of interferons, inhibits the growth of some microbes, and speeds up body reactions that aid or repair. That bolt right there tells you why we do this. Okay? It's not a bad thing to a certain degree. When temperatures go really high, though, fever gets too high, then things can be an issue. But slight fever actually does benefit us to a certain degree. It actually increases the performance of our immune system. At the onset of a temperature, an elevated set point will cause the hypothalamus to initiate sympathetic signals, causing vasoconstriction of peripheral blood vessels in the skin in an attempt to prevent heat loss. This does not help conserve heat loss. We'll shiver. This is why you get the chills. Healing tissue cells stop releasing chemicals, which then causes the set point to return to normal, triggering any process that causes us to cool our body. One of the most common things that we do to cool our body is sweating. So this is why a lot of times when your fever breaks, you wake up and you're soaking wet. Aspirin, like most of the drugs, have many actions and many uses. It is described as an antipyretic, which pyrogen means to cause a fire or fever. So that's why it can lower fevers. You also see aspirin described as an analgesic. Algia means pain, so it's a pain reliever. It's also an anti-inflammatory drug, what we call NSAIDs. It can be used as a blood thinner. We mentioned previously that it doesn't actually thin your blood, but it reduces your blood clotting property or abilities. It works by stopping the synthesis and release of prostaglandins, so that's why we call it a prostaglandin inhibitor. So, all stuff that is part of your innate, non-specific immune system. Okay, here's a nice summary of those various things. Now we get to talk about the really cool stuff. The adaptive immune system, which we mentioned the other day, has that memory component and will target certain things. Those certain things being antigens. Two properties distinguish adaptive immunity from innate: specificity for particular molecules, those molecules being foreign antigens involved. Unfortunately, distinguishing self from non-self, that sometimes doesn't occur. And I talked about how we can reject transfusions and that kind of stuff. And the memory for most previously encountered antigens, so that a second attempt prompts an even more rapid and vigorous response. All right, those T and B cells that we've talked about for the last couple of weeks are the two major cells involved with our adaptive immune system. There's actually two different forms of adaptive immunity: cell-mediated and antibody-mediated. T cells, which we call T cells, short for T lymphocytes, because they are derived from our thymus, T for thymus. They actually are produced in the bone marrow, but then, as we learned the other day, are going to develop that immunocompetence in our thymus. We call B cells or B lymphocytes B cells because they're produced in our bone marrow. So I think B for bone marrow. B begins B, cells, B begins bone marrow. Our T cells involved with cell-mediated immunity account for 70-80% of the circulating lymphocytes, become associated with our lymph nodes, spleen, and other lymphoid tissues. We talked about those the other day. Before leaving the thymus, they develop that immunocompetence, like going to basic training and getting trained to be a soldier. There are two major types of T cells that mature and exit the thymus. You have helper Ts and cytotoxic or killer Ts. The CD4 and CD8 abbreviations there relate to kind of their more biochemical classifications. But helper Ts, you can, based on their name, tell what they do. They help activate other T lymphocytes or even B lymphocytes to become plasma cells. We'll talk about plasma cells here in a second. Cytotoxic or killer T cells combine with the antigen on the surface of the foreign cell, causing lysis of the foreign cells, release of cytokines, the chemical messengers that we've been talking about. Lysis means to break down, so they help to break down those foreign cells. Other T cells include memory Ts and regulatory Ts. Memory Ts remain inactive until future exposure to the same antigen. If they encounter the same antigen at a later time, they're going to respond quickly due to the presence of thousands of them. And regulatory T cells provide a break on the specific immune response by inhibiting killer T cells and B cells, prevent against diseases caused by the immune system like allergies and autoimmune diseases. Essentially, they prevent the immune system from going haywire and attacking itself. B cells from the bone marrow are going to produce an antibody-mediated immunity response. These account for the 20 to 30% of our lymphocytes and also become associated with our lymph nodes, spleen, and other lymphoid tissue. When they become sensitized to an antigen, they will proliferate and differentiate to form clones of daughter cells that either produce antibodies specifically against the antigen or become memory B cells. So these B cells have the capacity to develop into one of two cells. They can either become a memory B or they can become plasma cells. Plasma cells are the ones that produce those antibodies that we mentioned the other day, those immunoglobulin proteins. Okay. Memory Bs can actually eventually become plasma cells, so that's their kind of function. Let's talk here a little bit more about cell-mediated. We'll get back to talking about Bs and those others just a little bit. Cell-mediated immunity particularly is effective against intracellular pathogens. So these are pathogens actually within our cells. They're also effective against cancer cells and foreign tissue transplants. So if you get like a heart transplant or a liver transplant, fortunately these guys are, or this type of immunity is going to try to combat that transplant. It begins the activation of a small number of T cells by a specific antigen. We then produce clone cells that recognize that same antigen as the original lymphocyte. Some of these clone cells become effector cells, while others become memory cells. Effector cells carry out the immune responses that ultimately result in elimination of the intruder. It's a lot more complicated than this, as you guys can imagine. I'm kind of trying to keep it pretty simple for you. Same thing for antibody-mediated, which, as you can see, works against extracellular pathogens. Pathogens found outside of cells, so includes bacteria, viruses, fungi, and our body fluids. Since we're talking body fluids, the other name for this is humoral immunity, because it happens in our humors Humors is a fancy way to describe, in this case, body fluids. As I mentioned, B cells are the ones involved with antibody-mediated immunity. B cells can transform into plasma cells, which are the antibody factories of our body. Antibodies, we learned, are proteins that circulate in our blood and other body fluids. Each B cell produces only one specific type of antibody protein. What that means is an antibody that can bind to only one specific antigen. When an antigen binds to its antibody receptor on the B cell surface, it stimulates two things: mitotic division of B cells, so division of and formation of more B cells, and the specialization of those newly formed B cells into plasma cells and memory Bs. Plasma cells, like I said, factories for production of antibody proteins. A few days after exposure to an antigen, a plasma cell will secrete hundreds of millions of antibodies each day for about 4 to 5 days until it dies. Memory cells do not actively participate in the immune response to the antigen or the initial immune response. Instead, they can quickly proliferate or divide, differentiate, and then become more plasma cells, as well as more memory B cells should the same antigen reappear at a future time. It's important to understand that these two divisions of the adaptive immune system don't work independently of one another, which is demonstrated down here at the bottom, stating that helper T cells aid both types, and the two types work together. And we can see that here on this image. In most cases, when a particular antigen initially enters the body, there's only a small group of lymphocytes, helper Ts, killer Ts, and B cells with the correct antigen receptors to respond to that antigen. Depending on its location, a given antigen can provoke both types of adaptive immune responses due to the fact that when a specific antigen invades the body, there are usually many copies of that antigen spread throughout the body. Some copies of that antigen may be present inside body cells, which will provoke that cell-mediated immune response. This is what we're showing off with our T cells here. And here's our two major types of T cells: killer Ts, helper Ts. And you can see the helper Ts not only help the killer Ts, but also the Bs. And they form those clones, and the cytotoxic or killer T cells will protect us against those intracellular pathogens, but also cancer cells and tissue transplants. We already mentioned that. Other copies of the antigen may be present in our extracellular fluid, which would provoke the antibody-mediated immune response, which involves those B cells, which we've got from B cells, either memory Bs or the plasma cells. The plasma cells, once again, form the antibodies, which we'll talk about what antibodies do here in just a second. This illustrates that cell-mediated antibody-mediated immune responses often work together to eliminate the large number of copies of a particular antigen in the body. All right, let's talk more about these antibodies. We learned the other day that they are from the protein class gamma globulins. They're made up of 2 short chain or light chains and 2 long or heavy chains. And you can see those down at the bottom. I put a very small image there. It's not a very important image by any means. But you can see the light chains and the heavy chains are labeled. Okay. There's also, and you can see that a little bit better over here in this part, an antigen binding site. Okay. The antigen binding site does exactly what it sounds like. It binds to the antigen. And binding of the antigen is very specific, which also activates the production of more antibodies specific to that antigen. Okay. There are 5 classes of antibodies, with each having a specific or slightly different role. So we abbreviate them IgM, IgA, IgD, IgG, IgE. The Ig stands for immunoglobulin. So we have an M, A, D, G, and E class. Okay. The IgM class fixes the complement system and are found in high numbers when the body first encounters an antigen. IgA class, found mainly in our mucus of the nasal, salivary, lacrimal, bronchial, intestinal, and vaginal secretions, blocks the entrance of antigens through those various places into our body. IgD is important in the activation of B cells. IgG is the most abundant and is very specific for its complementary antigens. Something cool about it is it can cross the placenta. What's the placenta? It's a connection between mom and baby. It's how baby gets nutrients and removes wastes. It's also how baby develops their immune system, this adaptive immune system, even before they're born, which is very cool. Mom can even give some of her adaptive immunity to baby before the baby's been exposed to anything outside of the body. Okay. IgEs are involved in allergies, so those are the ones that give you those allergies that we all love so much. They also aid in immunity against parasitic worms and other parasites. Okay. As a whole, what antibodies can do are illustrated here on this page. Okay, they can neutralize antigens. What we see there in our figure, letter A, what they do is neutralize some bacterial toxins, prevent attachment of some viruses to body cells. So we're showing the little Y-shaped, kind of blue-colored structure is the antibody. Little red triangular structures are the bacterial toxins that are neutralized. Letter B shows the agglutination and precipitation of an antigen. Okay, they basically cause them to kind of be stuck together, which then allows for those Pac-Men to come around and phagocytize them. Okay. That also is illustrated here with their ability to immobilize them, okay, meaning that they can't move, which makes them easier to be eaten. They also enhance phagocytosis by acting as a flag to attract phagocytes, which is what we see down there with letter E. And then they also, letter D, show how they activate the complement system. Okay. When the microbe is bound with an antigen, it activates the complement protein, whereas before it was inactive. So those are the 5 things that antibodies can do. Table 22.3 is a table from your textbook showing you not only their structure and their classification on the left here, but gives you a little bit more about their characteristics and functions on the right. I didn't get into them in super great detail, but you can read through that on your own if you want to. That's not something I'm going to pull anything directly from for the exam, but there's a lot of good information in there. Associated with antibodies is this concept of primary and secondary response. When a person is exposed to a particular pathogen for the first time, there's going to be a latent, we've seen that word before, latent means a delay. So a delayed period of 5 to 10 days before we see measurable amounts of antibodies appearing in our blood. Okay, we can see that here at the bottom. Okay, we're first exposed, about a week or so goes by before we see, and we're using G's and M's here as our illustration before they start to go up in number. This sluggish response is termed the primary response and is maybe not enough for a person to be prevented from getting the disease. But look what happens when they are exposed a second time. A secondary response is immediate, basically. Less than 2 hours before we're seeing high levels of antibody production, which should be enough, usually is, to prevent one from acquiring the disease. So that's great. And the immunological memory provides the basis for acquiring immunity against pathogens. This is why we get vaccinations, so that when we encounter something like the chicken pox, our antibody production can be immediate, and we're not going to catch it. You guys are probably at that age where some of you guys had the vaccine and some of you actually had it as kids the old natural way. Anybody know if you had chicken pox as a kid? A couple people can. You guys might not be aware of this, but back in the day, when there wasn't a chicken pox vaccine, one of the things that parents like to do is just get it over with their kids. You ever heard of a chicken pox party?
Yes.
Chicken pox parties are pretty crazy to think about, but it used to be when a kid on the block would get the chicken pox, mom and dad would say, you know what, let's get our kid the chicken pox. They'd send their kid over to play with the infected kid. Why? So that they could be exposed that first time and get the chicken pox. Chicken pox is something that is much more deadly the older you get. Just get it out of the way. Fortunately for many of you guys, sounds like, or looks like, you guys didn't have to have that because, well, you had the vaccine. Okay? Vaccines, I cannot stress enough, are super, super important. Okay? Do not fall victim to the belief that vaccines are dangerous. They are not. They are absolutely the opposite of that. Okay? So get yourself vaccinated and your kids vaccinated. All right. Looking at the ways that we can acquire immunity or adaptive immunity, there's two kind of principal ways, active and passive. Active involves that vaccination that I was talking about, is one of the ways that we can do it. And this is conferred when the body manufactures antibodies in response to direct contact with the antigen. Okay, and then when they're exposed to it again, the body remembers it, mounts a quicker and more specific antibody response. Okay, active immunity can be conferred by getting the chicken pox or by artificial injection of it. Okay, which a vaccination is going to include dead or weakened pathogens. I always think you have to be active to go out and get a vaccination. You have to actively move your body to go get that. You have to actively catch the cold or whatever it is that you're catching antibodies for. Passive is conferred by the transfer of antibodies from person to person. What that means is the person that is receiving them doesn't actually produce their own. An example of this was when I was talking, mom can deliver the IgGs across the placenta to the fetus before the baby's even born. All right, last thing I want to talk about are things that involve dysfunction of the immune system. Collectively, we have several different types of autoimmune diseases. I'm only going to talk about some of the more prominent ones. So, defining autoimmune diseases, these are diseases which occur when the immune system fails to display self-tolerance, attacks the person's own tissues. Some of these we've seen before. Okay. Multiple sclerosis is where the white matter of the brain and spinal cord are destroyed. Okay, so that's the myelinated axons that we talked about previously. It's going to result in a whole bunch of issues. Myasthenia gravis impairs communication between nerves and skeletal muscle. Mentioned that one previously a little bit. Type 1 diabetes, we learned, is where the beta cells in the pancreas are destroyed, therefore the person cannot produce their own insulin. Rheumatoid arthritis destroys joints. Okay, who gets rheumatoid arthritis? What are you saying, Caleb? Say it. You're actually incorrect. Grandma and Grandpa get osteoarthritis. And that's what happens as you get older. Your joints break down. Rheumatoid arthritis, which is autoimmune, actually happens a lot more in kids. So this is a debilitating disease. Kids should not have bad joints, but rheumatoid arthritis is an issue with them. Not to pick on you, Caleb. You're one of many that has answered that incorrectly over the years. So, Graves' disease. We've mentioned this before. It's antibody-induced stimulation of the thyroid gland. Can cause all sorts of different issues. Therapies and treatments for various autoimmune diseases include removal of the thymus gland, injections of beta interferon, immunosuppressive drugs, and then a process that's called plasmapheresis. This is a pretty cool process where the person's blood plasma is filtered, then the antibodies and any antigen antibody complexes are removed. So it basically just tries to slow down the role of the immune system. And it's something that we all suffer from are allergies, which is where a person is overly reactive to a substance tolerated by most other people. The other term for allergic is hypersensitive, and the things that we are allergic to are called allergens. This is kind of the time of year where a lot of people suffer from seasonal allergies as stuff starts to bloom and be released into the air. I'm very lucky that I don't suffer from seasonal allergies like my wife. I hope my children are like me in that regard. I also hope my children are not allergic to cats like I am, and they completely knock me down. So it's never fun when I'm exposed to a cat like at my father-in-law's house. So I have to be doped up on allergy meds. Now, there are two types of allergies: immediate hypersensitivity and delayed. Immediate is caused by abnormal B cell response to allergen, and it's within seconds to minutes. Like me when I walk into the house, my father-in-law's, I'm within minutes sneezing and eyes watering and all sorts of other fantastic stuff. Okay. Delayed is related to abnormal T cell response, and this is much longer after exposure. Anyway, think of a situation where a person gets exposed to something and then a couple days later they notice it. You might experience this if you're hanging out in the woods. You brush up against poison ivy. Does this. Most of us are hypersensitive to poison ivy. Allergies involve abnormal release of antibodies of the IgE class, which are responsible for producing a number of chemicals when bounded to the antigen. Chemicals that include histamine, which is why we take antihistamines, but also leukotrienes, prostaglandin D, as well as others. And here we can see a figure that illustrates what happens when the allergen binds to B cells. We produce plasma cells that produce the antibodies. Those antibodies involve mast cells that release histamines and other chemicals that cause things like your nose to run and you to sneeze and you to itch and so on and so forth. Any questions? All right.